Welcome to BABE project. BABE stands for Bird, Bat and Ant Exclusion. We conducted that project at 6 canopy cranes, spanning from Leipzig through Japan, China, Papua New Guinea, all the way to the South Wales in the southern Australia. We were working on these cranes where we were excluding the predators from the canopy by the cages which were protecting the trees against the vertebrate predators, and we also excluded the ants by sticky traps, which we sometimes combined with the vertebrate exclusions, and sometimes we conducted them independently. So we had a fully factorial predator exclusion experiment. We worked in the canopies and in the understories, and altogether we worked on 480 small trees in the understory, which were high up to 3 meters, and 480 trees branches in the canopy at each of the study sites. At the beginning of the experiment at each of the study sites, we collected all the insect, and then we resurveyed the insect later on, 4 months later, when the effect of the predator exclusion was measured. We collected the insect alive or to ethanol, and later on we identified all 2 the morphospecies, or at least 2 feeding guilds for the groups which were difficult. With the live insect we focused on several groups, for example on galling insect which we reared to adults, and we will work with the data later on. We also did some feeding experiments with the insect which we kept alive and identified how much herbivory damage they are causing. To measure the herbivory damage in the understory and canopy, we focused on the change in the herbivory. That's why we numbered the leaves at the beginning of the season when the leaves were flashing, and we scanned them later on, and we identified how much herbivory damage occurred in the predator-free and predator-accessed branches and trees. We also surveyed the predators. We baited the ants on sugar and tuna, and we surveyed the ant communities, and we also surveyed the bird communities. Sometimes passively by the recorders placed in the canopy and understory, and sometimes actively to the mist nets where the permits allowed. From the birds which we captured, we were obtaining various samples, but mainly we were focusing on what they eat. So we were making them vomit by emetic tartar and identifying what diet they were consuming before. Similarly, we focused on the bats, and again we sampled various samples from them, identified what they eat through the stable isotopes, and we always released them happily. From the birds, the diet was identified under the microscope and then through the sequencing. With the leaves which we collected, we conducted various chemical analysis. We measured the traits, physical and chemical on them, and identified how they are responsible for the herbivory damage. The experiment was sometimes fun and easy in sunny days, but sometimes quite challenging in the windy and harsh conditions. There is much more which will be coming out of this project and a massive dataset, so please follow us and keep an eye on the new results.